Okay, now we're going to move on to activity 3 in exercise 13, ecology. Activity 3 is looking at community variance. I'm going to go ahead and share my screen. So community variance, ecologists often need, okay, so first of all, what was in a community again? So remember, a community had 3 parts, and if we go all the way back, sorry about this craziness here. So a community had 3 important aspects. It had feeding interactions, which we already discussed in activity 1. It had symbiotic relationships, which we discussed in our introduction, and it has biodiversity. So when we looked at biodiversity, there was 2 components, richness and evenness. So now we're going to look at community variance. So ecologists often need to estimate the biodiversity within communities. To do this, samples of the number of species and the number of individuals must be taken in each community to be compared. So the best way to do this is often causes a lot of debate, but to do this, we can sample using transects or plots. I will discuss both of those a little bit more in the next slides. But when we do use transects or plots, we want to look at both richness and evenness. Remember, this contributes to biodiversity. And your job is going to be to use the photos on the last 3 slides to compare the biodiversity between the 3 sets of communities. You're going to fill in the table in your lab manual for each photo of a community, noting the number of species, richness, and relative abundance of each, evenness. Then answer the questions on your worksheet. So community variance. So we're essentially asking what type of organisms are present and how many are present. So there's 2 ways to measure. There's transects or plots. So just going back to biodiversity very quickly, it has 2 components. We have richness and we also have evenness. So richness is looking at the number of species. Remember, the more number of species we have, uh, if you have we're looking at number of species. If you have more number of species, it's more diverse compared to something that's less rich, it's less diverse. When we look at evenness, we're looking at how much of each species is present. So when you have more evenness, you have a more diverse um, community than if you have just one species has there's more of that particular species compared to the other species. This is less diverse. Okay, so transects. In a transect measurement, an ecologist will follow a line and take data along the length of the line. So here in the green square represents our community. This red arrow is the transect, and the ecologist would take the data along this specific line. Sometimes it will be in various intervals, but as shown here. So here you have your community. The line is shown here in red, and then it's taken at various intervals. So 1, 2, 3, 4. I don't know what the space between each interval is, but let's just pretend it's 1 meter interval or something like that. But it could be taken at intervals as shown here, or it can be continuous along the length of the transect. Another way you can take this data is using plot measurements, which involve counting all the individuals within some area. So as you can see here in the left, it's really easy to count the um, different species you see, the different trees you see. So A, B, C, and D in community 1 and community 2. But imagine if you were looking at a forest, this would be very difficult to do. So ecologists can take a community, which is represented here by the green square, and they will put a plot somewhere, which is represented here as the red circle. This plot can be either a circle or a square. It depends on the experimental design. And the size of the plot may be very large, very small. This also depends on what species is being counted and the methods and research design that the ecologist is using. But essentially, the ecologist will count all of the individuals within either the circle, the plot, or the square, the plot within the community. So if we were on campus, you would have gone out to the RCC um, campus, found a desired community and placed a hula hoop down with your lab members, and you would have counted the evenness and richness of that community. Um, Instead, you're going to look at these next three slides and determine the evenness and richness for each of these communities. So comparison number one shows community A, B, and C. Comparison number two shows community A and B. Comparison number three shows community A and B. Um, so for here, you probably do not want to put down any kind of plot or transect. This already looks like a snapshot of that or of the community. Here, you can decide if you want to put down a plot. You probably do not need to. As you can tell, you probably don't need to do it here either. And then here it's already done for you. So just work through those problems. I'll put the worksheet up and the quiz up as well. Oh, and I just want to mention I did put an extra credit opportunity up, so please go ahead and take advantage of that. If you are missing any assignments, please contact me so we can make sure you get as many points possible before the semester ends and you are and your final practical happens.